Thank you. I am in the Division of Microbiology and Immunology, so like microbiologist by association. So something I really enjoy as a faculty member is taking meetings in the offices of my colleagues. These are really informative conversations, and I find it really interesting the objects we choose to surround ourselves by at work. I'd like to invite you all to my office someday. And if you came there, you would see I have a number of plants, artwork, pictures of family and friends, and I also have this clock that sits on my desk that I received on October 22nd, well, in commemoration of October 22nd, 2013, which was a day that changed my life, both personally and professionally. So, as mentioned, I'm Aaron Petrey. I'm an assistant professor in the Department of Pathology, the associate director of the molecular medicine program, and I'm here today to tell you about a type of molecule called a glycan that is able to link inflammation and blood clotting in diseases like sepsis, COVID-19, and inflammatory bowel disease. So the story behind my science really begins when I earned my PhD at the University of Georgia in the labs of Richard and Heather Steet. There, at this complex carbohydrate research center, we studied a class of molecule called glycans. And these molecules were central to the pathology of children with a rare pediatric genetic disease. Now, these patients had a syndrome of disorders, and the most severely affected of them did not make it past the first decade of life. We really want to understand, well, what caused the underlying pathology. And we used cells from these patients as well as zebrafish to study the early developmental effects. Now, as a first-year graduate student at the time, I knew the central theme of molecular biology, and that's that DNA is transcribed into RNA, which is translated into protein. Proteins make cells, and cells make organisms. But what I learned in my PhD is that life, health, and disease are a lot more complicated than this little textbook linear process. Many molecules go into health and disease, and in the patients we studied, glycans were the underlying factor. It was not a protein. So this is an image of cells from the patients we studied. And these glycans accumulated inside of their cells, and you can see them in these little black dots here, and they caused a whole host of disorders, including chronic inflammation. Now, you may think you've never heard of glycans before, but they actually define the blood group system we use at the hospital. For those of you who are type A in the audience, you make one type of glycan. I'm type B, I make a different type of glycan. If you're AB, you happen to make both of those. And if you're a type O individual in the audience, your blood type's defined by the fact that you don't make type A or type B. So the concept that these glycans could have critical impacts on inflammation and chronic disease motivated me to understand the underlying biology. And I took my postdoc at the Cleveland Clinic in the laboratory of Carol D. Lamotte, a glycobiologist who studied these glycans, and with Claudio Fiocchi, my clinical mentor, who is a gastroenterologist. And there I trained in bench-to-bedside research, where we would bridge the work in the clinic and the laboratory to understand the underlying causes of disease in patients who really had an unmet clinical need. Now, that unmet clinical need was an inflammatory bowel disease. This disease consists of two types of disorders. Crohn's disease, which is characterized by lesions throughout the gastrointestinal tract, and ulcerative colitis, which is predominantly confined to the colon. Now, there are some similarities between these two forms. They both lead to lifelong, relentless destruction of the GI tract. And both of these groups of patients experience a 16 times increased risk of blood clots. And in half of the patients who get a blood clot, those can be fatal. Now, these are lifelong disorders with no cure. We do not understand why patients get them. We do not understand why the incidence of these diseases are increasing in the United States and globally. There are about 3 million Americans affected by IBD, and the annual healthcare costs amount to about $8.5 billion. And while there are a number of increasing therapeutics on the market to help treat patients, about 40% to 50% of patients fail therapy, underscoring an unmet need, and none of the therapeutics actually address this risk of increased blood clotting in these patients. Now, only 6 months into my postdoc, this is October of 2013, I received a phone call that I was a bone marrow match for a stranger who needed a bone marrow transplant. And I flew to Washington, D.C., and you can see me there holding a bag of bone marrow stem cells and a bag of platelets. And as I was holding these, over the next couple of months, I really started thinking about this process, and it changed my life, and it changed my scientific career. And I started wondering, these patients have these defects in platelets, platelets are born in bone marrow, and could platelets be an overlooked driver of disease in the patients we were studying? That brought me here to the University of Utah, December 1 of 2019, where I joined the Department of Pathology and the University of Utah Molecular Medicine program, where we're world-renowned for our research on platelets. Investigators here at the University of Utah changed the way the world thinks about platelets. These cells were only thought of as playing roles in blood clotting, but we now know they talk to many cells in our blood, including immune cells. And platelets have the capacity to either help us improve when we're sick or make us worse when our disease might be chronic. So platelets are these small sentinels that circulate in our blood. They're the second most numerous cells in our blood. And they respond to danger signals. So in the case of like an injury, a cut, or an infection, maybe seeing a virus or bacteria, these platelets can activate. Once they activate, they release molecules. Those molecules can then participate in blood clotting or stimulating the immune response. And I started to wonder, if platelets are these sentinels and they can respond to injury and infection, could they be responding to glycans in our patients, and could that be contributing to their disease? This is an image from one individual. These are 2 biopsies. So on the left, this tissue is not affected. By biopsy, when the patient gets an endoscopy, this is going to look totally normal. But on the right, that tissue by biopsy would look incredibly diseased. And in red, that color labels a specific glycan structure that can recruit platelets and immune cells into inflamed sites. What I want to draw your attention to here is actually on the left, in the non-affected region of tissue. We see this glycan building up where these arrows are pointing. That's a blood vessel. So what this told us was that these changes in glycans occur early in disease and non-affected tissue. That'll eventually become just as diseased as on the right. And we thought platelets could interact that early on. So through a number of studies, we were able to demonstrate just that early on in inflammatory bowel disease, platelets can actually protect us from inflammation. And that's what they do in many healthy individuals. But individuals with IBD, this process goes awry, and the platelets instead contribute to increased inflammation in their disease. These studies led to the cover of a prestigious journal, Blood, and we were also further honored by being named Cover of the Year in Blood. But we really wanted to go a bit further. And in collaboration with John Valentine here at the U in the Division of Gastroenterology, we were awarded a grant by the National Institute of Heart, Lung, and Blood to understand this process. And so our team identifies and consents patients with IBD. We collect blood from patients, and then we measure how do their platelets respond to those little signals of injury or things that activate them. What you can see in the top right is that patients with IBD, their platelets are more likely to form clots very rapidly compared to healthy individuals. And then below that, we compare each individual that we get in the clinic, and we measure their RNA, so that's in columns, and the different little colors, whether it's red or blue, indicates changes in certain molecular pathways we think are very important in IBD. What we've been able to do with this knowledge is identify a pathway in these patients. This is a receptor that recognizes a glycan, and it contributes to increased clotting in IBD patients. Now we can use currently existing drugs, treat platelets with them, and we can try to recover platelet function in IBD patients back to normal levels. So they should hopefully still form clots and not cause a bleed, but not form clots when they shouldn't and cause disease. So over the past about 11 and a half years, this experience where I donated bone marrow really changed my life, and it led me into places I really wouldn't have expected to go scientifically. It brought me here to the University of Utah. And actually my first month as faculty here, so it's December 1 of 2019, I connected with the recipient of my bone marrow, Pamela. And I learned that she's an avid watercolor painter, and she's had about 11 more years of life to spend with her family, see her sons get married, and she's continuing to paint watercolor portraits like this one still to this day. And this is one of my favorite ones. It reminds me of our beautiful state. So none of this would be possible without the support of my partner, Kimberly, who helped me build our laboratory, the individuals in my lab who give it their best every single day, our clinical collaborators, my department, my division, the molecular medicine program, and importantly, the National Institute of Health that funds us, and for all of you for supporting our work. Thank you very much.